I'm Rocio Sancho.
My name is Sergio Pedraza.
I'm Cristina Garrone.
My name is Teoni Demcelari.
Our work is focused on trying to find new cures for type 1 diabetes. Over the last decade, we have witnessed a great advance in the development of beta cells from stem cells. In my lab, we want to understand how this process is actually controlled, and we want to achieve more functional beta cells and more yield of beta cells from stem cells. And we work on a project funded by JDRF where we are trying to explore the use of 3D growing culture conditions to make these two things possible.
The pancreas has two different parts. One is involved in the digestion, in the regulation of the digestion, and another one is involved in the hormone release. In this part, there are different types of cells, and one of them is the beta cell. Its function is basically produce and release insulin to control the glucose levels in the blood.
They're very sociable cells, and in fact, to function properly, they actually require cell-to-cell contact so that they can secrete a synchronized wave of insulin.
The most interesting thing about beta cells for me is how they sense the glucose levels in the blood. It's a quite complex process. It involves a lot of enzymes and molecules, but I think I found it really interesting.
So I was always interested in diabetic research because I have close relatives who are afflicted by the disease. So I wanted to be part of the team that would shape new therapies and improve the quality of life of patients.
I found it really interesting because I want to improve the process of training beta cells. So I think this research may be really useful to achieve that and to improve the lifestyle of the diabetes patients.
So in the lab, we have found different growing conditions that allow expansion of intermediate cells that give rise to beta cells. And we also have found conditions that will make those cells more functional. So the next steps is to actually combine efforts with different groups in order to address whether those cells are really fully functional, and then to combine efforts with the medical community in order to make or translate our findings to a more clinical setting. Eventually, what we think is that this research that we are doing, funded by JDRF, will allow the translation of stem cell or beta cells derived from stem cells into patients in order to cure diabetes.